pleasure to introduce Maddie here, who arrived at Davis pretty recently, got her PhD at the University of Toronto with Marty Kurkosek, and did a lot of great work that you're going to hear about on disease dynamics. And what's super cool about it is how it spans across human and agriculture and wildlife diseases and brings a lot of different threads together across those and brings so many different new quantitative tools across statistical and dynamical modeling. And as Maddie has started here at Davis, she's working with both me and Alan, bringing a lot of the concepts from her thesis on early warning signals and decision support modeling to whole new areas and in exciting and innovative ways. And so we're going to hear about some cool science, and take it away, Maddie.
Thanks, Marissa. I'm really excited to talk to you all today about some work I did during my PhD, because that's the work that I've most recently finished. So looking forward, hopefully, to some talk in the future with new work. But anyway, okay. So I think parasites are really, really interesting. I hope a lot of you are already in agreement with me, but in case you're not, I think they're really cool, but they're also really important. So parasitism is the most common consumer strategy. Parasites represent 40% of currently described species, and despite their often small size, account for really significant biomass. They're important for regulating host population dynamics and are involved in a large proportion of trophic interactions. Because of all these interactions that they contribute to and shape, they have really large impacts on global health and economics, if you remember a recent pandemic. And like everything else that we love and study, they're also impacted by global change. And there's a lot we don't know about how climate change will affect disease systems. We used to think, for example, that climate change would make for a sicker world. And we were, and I guess still are, really interested in the impacts of climate warming on vector systems and how warming might increase vector-borne disease incidence by increasing vector range. But like, there's still debate around how public health interventions can mask climate effects on these types of systems, making their impacts very difficult to track. It's also true that people are worried about the impacts of climate change on the frequency and intensity of extreme climate events and how that might increase disease prevalence, particularly in marine systems. But it's also been very difficult to establish a kind of baseline for disease in marine systems. So again, open question. And finally, there's kind of increasing interest in the impacts of climate change on parasite extinction rates. So arguably in the midst of a mass extinction event. And parasites are subject to those impacts as well, because so their livelihoods, not really their livelihoods, but their abilities to persist are really dependent on, obviously, their hosts. These kind of co-extinction events are of increasing interest, and because of their interactions with all of these species, parasite extinctions could have really important impacts on ecological systems. But I guess what I'm trying to get at here is that there are a lot of gaps in our knowledge, and I think the consequences of these gaps remaining unfilled are potentially quite large. So it's important to figure out which questions we should ask and how we should ask them and how we should answer them. So I'm going to talk about a few projects where I try and contribute in a very, very small way to some questions, some open questions in disease ecology, and raise some questions about maybe where we should be focusing our time and money, things like that. So first, I'll talk to you about the impacts of behavior or of parameter and behavioral uncertainty on the estimability of within-host population dynamics models. And then I'll talk a little bit about the impacts of environmental variability on within-host diversity and among-host transmission. And then finally, I'll talk a little bit about early warning signals. This kind of gives away the game, but if we can use them to predict a warming-induced experimental epidemic. Okay, so project one. Okay, so at the within-host level, these mechanistic models that we use to represent within-host population dynamics often draw from consumer resource theory, and namely Bloch-Volterra models, which describe the population dynamics of interacting predator and prey species. and have been used to describe a really wide array of consumer resource systems. And when applied to within-host disease systems, it's often posited that the host immune system behaves analogously to a predator, and that parasites behave analogously to prey. And that functional responses, which were originally developed to describe a consumer's response to resource density, can also describe an immune system's response to parasite density. So for example, at very low prey densities, it might take predators a while to learn how to consume prey items or even find prey items, creating this kind of lag in consumption that we see here. And similarly, it's been proposed that a novel parasite may induce a learning time in a host immune system. It may take a while for a host immune system to develop an adaptive immune response and actually get this whole interaction going. And similarly, at high parasite or high prey densities, predators may be limited in their ability to consume prey items, both by their handling time or just number of prey items available, producing this saturating effect. And analogously, we might consider the time it takes immune cells to actually dispense with parasites, and that has been estimated in empirical systems a few times, which is kind of neat. So this kind of conceptual underpinning has been used to build mechanistic models of pretty highly specific within host interactions, and I've included a few examples here. But what about poorly understood systems? So within host systems can be tricky because it's not always clear which components of the immune system and the infection process that we should be modeling or measuring. And I think this presents a use case for kind of generalized version of a within-host population dynamics model. And there is precedent for this. SIR models, which I think you're probably all familiar with, are a kind of generalized version of a model that describes how a disease moves through a population of hosts. And we can use this kind of non-specific SIR model and add some details when we know a little bit about a system to describe a disease system fairly quickly. And again, as we learn more, kind of elaborate on that general form. And importantly, these kind of general SIR models have been evaluated for estimability and have been used to estimate key parameters during outbreaks, like transmission rate and R0, which you're probably familiar with as well. That's the basic reproductive number, and I'll talk a bit more about it later. And while a few generalized models of within-host population dynamics have been developed and proposed, they have yet to be evaluated for estimability. In other words, we haven't figured out if fitting these models to data can actually provide us with reasonable and reliable parameter estimates, which kind of raises questions about their utility when confronted with data. So to kind of address this, I used a couple different general within-host population dynamics models to assess the reliability of parameter estimation, the consequences of misassigning the underlying mechanistic model, and the reproducibility of observed dynamics. So we started with these two, again, non-specific models from the literature, which I'm just going to call Model 1 and Model 2 for ease here. And they both track parasite and host immune cell populations through time, as represented by P and I here. And in both models, the parasite grows by some intrinsic growth rate, R, and shrinks as it's consumed by the host's immune cells. In Model 1, that consumption is dictated by a single parameter K, and in Model 2, consumption can be additionally mediated by the immune system's handling time. And in both models, the host immune system grows in response to infection. In Model 1, that growth is proportional to parasite density at low parasite densities and saturates at high parasite densities. And in Model 2, the growth of the immune system can be shaped by one of these three functions, which we can just call type 1, type 2, and type 3 responses to parasite density. So next we use these generating models to simulate demographically stochastic time series. And per generating model, we simulated 5 stochastic time series. And we can read these as time series plots with time on the horizontal axis and population abundance on the Y. And I'm showing parasite populations here in green and host immune cell populations in blue. And I'm showing deterministic simulations as solid lines and stochastic simulations as dashed lines here. So if we take a look at this first model, we can see the host immune response growing in response to infection, the infection coming down as parasites are consumed, and then eventually the parasite is cleared from the system. And when the immune system, if we take a look at model 2, the immune system, if it has a type 1 response to parasite density, the second model shows this initial growth of the parasite population, a subsequent immune response, which is larger, before each population enters and settles around some kind of stable equilibrium value. In the same model, when the immune system has a type 2 response to parasite density, we can consider a few different parameterizations of the model that each produce different dynamics. So first, we can consider a case in which each population experiences damping oscillations towards some equilibrium value that's not shown here. Second, we can consider a case or a parameterization, rather, in which both populations establish stable oscillations. And third, we can consider a case where both populations exhibit divergent oscillations. Finally, if the immune system has a type 3 response to parasite density, the second model, again, shows the initial growth of the parasite population, the response of the immune system before both populations settle into some equilibrium. So the second model here is more so showing a kind of chronic infection as opposed to an acute infection in which the parasite is extirpated relatively quickly in model 1. So with our generating models and stochastic time series in hand, we fit each generating model to each time series. So the generating models here on the left and the time series are listed on the right here. We did this in 2 different frameworks. So we simulated a continuous time version of the model and a discrete time version of the model. So we fit the models to the data in global and autoregressive frameworks. So for example, this first model here, we fit to 5 matching time series shown in red and 25 mismatching time series denoted by the gray arrows. And fitting the generating models to matching time series went okay. So here, each number denotes the number of cases out of 5 in which fitting the generating model to a time series provided us with some useful output. And then the different colors show us how often those useful outputs reproduce expected dynamics. So green colors mean that every time we got a usable output, parameter estimates reproduce system dynamics. Yellow means that in some of those cases, we got the dynamics we expected to see back. And red, nothing good happened. Okay, so here, for example, we can see that we encountered some issues when fitting the second model with the type 2 functional response to matching time series. We got some usable outputs, but they didn't always reproduce system dynamics. And that's all with continuous time model. But while fitting generating models to discrete time series, that went not as well, I would say. So you'll notice some issues, again, when we're fitting this second model with type 2 functional response to matching time series in an autoregressive framework. In every case, we get usable outputs. The model converges, everything looks like it's fine. But in a lot of cases, we're not actually getting parameter estimates that make very much sense, and they're not reproducing the dynamics we expect to see. Okay, so next we fit the generating models to mismatching time series. And while we see a lot of numbers less than 5 here and a lot of red, which is encouraging, we also see a lot of cases in which fitting a model to a mismatched time series gives us some usable output and also reproduces system dynamics, which is a bit odd, not encouraging. And while we see that fitting generating models to mismatching time series in a discrete time framework also produces usable outputs, we see that those outputs don't often or don't ever reproduce system dynamics, which inspires like a little bit more confidence. So after fitting generating models to matching time series, we wanted to know how parameter uncertainty affected our ability to reproduce these dynamical behaviors. So in fitting our generating models, when fitting the second model with a type 2 functional response to matching time series gave us some usable output that resulted in convergence. We drew 1,000 sets of parameter estimates from each posterior and simulated 1,000 new time series and determined what percentage of those gave us the behaviors that we expected to see. So when we fit the type 2 model to damping or divergent oscillations, we see more or less what we expected to see. A very high proportion of resulting simulated time series give us the behavior we expected to see, which is great. But when we fit that same model to data exhibiting a stable limit cycle, we find that parameter estimates don't do a great job reproducing that stable limit cycle. And I think that's just because the parameter space that produces this behavior is quite small. So it's really easy to move out of that parameter space and get something different. So either these damping or divergent oscillations. Okay, so we learned that fitting a misassigned model to time series data can produce reasonable parameter estimates that do reproduce system dynamics sometimes. Which to me is worrying because when a system is poorly understood, it might be quite easy to misassign the underlying model, particularly when data are oscillatory. Yeah. We also found that uncertainty in parameter estimation seems to really negatively impact our ability to characterize system dynamics, which really limits our ability to describe infection dynamics, especially when a system is poorly understood, and calls into question our ability to predict infection outcomes. So you can imagine the consequence of an infection that settles around some stable equilibrium would be maybe more manageable than an infection that's producing divergent oscillations, which you see in sometimes with malaria. Okay, so what can we do? It feels like this is maybe a difficult problem to address, but we did find that quantifying some key parameters does improve estimation. It might also be helpful to formalize a relationship between immune memory and handling time and functional response. There are some guesses in the literature as to what types of systems might produce which types of behaviors, but working on that theory a little bit more and connecting it to empirical systems might be helpful. You could also consider alternative approaches to model fitting or just dedicate a lot of time and resources to increasing our system-specific knowledge, especially for systems that we're really, really worried about. Okay, I'm gonna move on to project 2. So it's become really clear that climate and land use change can facilitate novel species interactions and in some cases increase contact rates. Oh, oops. No, I don't want to do that. No. Oh, I don't have a cursor.
Also, don't leave meeting.
Oh, wait, I have a cursor. I don't want to leave, but I don't want to do this either.
Wait, I think I can do it. There.
Okay.
Sorry.
Okay, cool. Thanks. Yeah, so it's become clear that anthropogenic change can increase contact rates between wild populations and humans, which really increases our risk of seeing these kind of zoonotic diseases coming to the forefront. It's also increasingly clear that within host population, diversity influences disease dynamics, but the mechanisms that promote within host diversity or multiple infection have been relatively understudied. The kind of baseline assumption has been kind of single parasite in a host, but we know that's not often the case. So we're kind of coming back now to this kind of classical question of why are there so many species, which is a great question that we continue to not be able to answer very well. But so in the 70s, we see the kind of emergence of the intermediate disturbance hypothesis, which proposes that at some intermediate level of disturbance, and level is defined in a bunch of different ways, we should see maximum species diversity. When we have really low levels of disturbance, we might expect to see competitive exclusion, and at really high levels of disturbance, we're just seeing a lot of extinction. And both of those scenarios kind of preclude species from coexisting for a long time. So a couple of decades later, we see the emergence of coexistence theory that proposes a number of mechanisms by which diversity can be maintained. We can kind of bin those mechanisms as being either equalizing mechanisms that reduce fitness differences between species, or as stabilizing mechanisms, which concentrate competition between members of the same species relative to members of different species, or inter versus inter-specific competition. So while there's this very long history trying to figure out why there are so many species, it's still unclear how these mechanisms will affect the promotion and maintenance of specifically non-equilibrium coexistence, or we're calling it here co-occurrence, especially within host disease systems, which are often characterized by multi-parasite infections and seldom exhibit long-term stable coexistence. So to address this question, we used a mechanistic model to explore the impacts of differently sized and timed environmental disturbances on within-host parasite diversity and among-host transmission. And there are a couple different reasons why environmental variability might be important here. So first, we can think about environmental variability as a feature of climate change impacting ectotherms. So we're seeing increasing variability over time in addition to increasing mean temperature. Second, bats have really long been acknowledged as prolific reservoirs for disease. There's some debate now on if bats are actually special or not. I'm not here to address that debate. But a lot of people think they're special among mammals because they have powered flight and they can control their internal temperatures as they move between torpor and powered flight. So they have these periods of very, very low internal temperature and periods of very high internal temperature. And it's been theorized that this kind of internal thermal variation may contribute to the efficacy of their immune systems and allow them to carry like a lot of microparasites at the same time without suffering increased morbidity, which is kind of this fundamental question in bats. So similarly, it might be interesting to think about what that thermal variability has to do with the number of microparasites they can simultaneously support. So we started by adapting the same model we started with in project 1 to include 2 parasites that indirectly compete through the host immune system, so through parent competition. And then we developed 4 sets of paired thermal performance curves to relate the temperature of the system, so the temperature within the host, to the replication rates of both parasites. And then we simulated that model forward in time under constant conditions and stochastic conditions, and then analyze the viability and longevity of parasite co-occurrence and transmission probability across a range of disturbance regimes. So you should be familiar with this model from project 1, but I'll complete it there. So this is the model that we first discretized and then expanded to include a second parasite. So we'll call the parasites P sub I and P sub J here. And again, those parasites indirectly compete through the host immune system. So I have a little schematic there. And first we considered kind of deterministic version of this model where all of the curves are smooth and beautiful and everything goes well. So second, we considered a demographically stochastic implementation of this model where birth and death processes are written as Poisson and binomial processes. And then we wrote an environmentally stochastic version of the model where each parasite's replication rate responds to the temperature of the system, which is highlighted right there, as dictated by different sets of Sharp-Schofield thermal performance curves, which I've shown here. And those curves each encode some advantage for one parasite over the other. So in the top right, for example, we can see that the average temperature of the system shown in pink should give P sub I an advantage over P sub J. Or sorry, this is supposed to be equal. The bottom left should give P sub I an advantage over P sub J. Bottom right should give P sub J a thermal advantage over P sub I. There are different ways to figure out how to give one parasite an advantage over the other. We tried a couple different ways, and we can talk about that later if you want to. And then finally, we considered a demographically and environmentally stochastic model, which combines both these demographic and stochastic processes with this kind of environmental variability here. And when considering environmental stochasticity, we implemented it via 195 different disturbance regimes that are differentiated by the magnitude and frequency with which disturbances occur. So we conducted a bunch of simulations of each stochastic model and treatment, a few of which are shown here, and used the resulting time series to ask a couple of questions. So first, under what conditions can secondary parasite PJ actually invade the system if P sub I is already in the system? And if the secondary parasite P sub J does invade, how long can these two parasites actually co-occur with one another? So deterministically, there is some condition for which we expect the secondary parasite P sub J to be able to invade the system. And depending on the parameterization you use, some co-occurrence is possible when the secondary parasite is introduced before the 10th to 12th day of infection, which we'll call our kind of deterministic threshold. And after it's been introduced, those two parasites can co-occur for some maximum amount of time. But once we incorporate environmental stochasticity, a different picture begins to emerge. So again, we can use our deterministic model here as a baseline, and that black horizontal line references the kind of deterministic case. And here I'm showing the demographically stochastic model outputs above these black sub D labels on the horizontal axis, and environmentally stochastic outputs above the blue labels. So S sub E is environmentally stochastic implementations, and S sub D includes both types of stochasticity. And above each kind of stochastic implementation here, we have 195 dots, each representing the outcome of a given disturbance regime. And the y-axis here is just telling us the difference between the deterministic implementation and the stochastic implementation. So each dot here is also an average of a bunch of simulations. So to summarize, we end up finding that with respect to that deterministic baseline, demographic stochasticity on average slightly shortens the amount of time for which these two parasites can co-occur. But once you consider environmental stochasticity, we see the potential for these parasites to co-occur for longer periods of time really emerge. And we can look into that with a little bit more detail with a heat map, which shows sizes and frequencies of disturbance on the vertical and horizontal axes, respectively. And the pink circles here denote our deterministic baseline for co-occurrence, and colors denote co-occurrence times relative to that deterministic baseline. So warmer colors denote longer periods of co-occurrence, and cooler colors denote shorter periods. We can talk about why later. We can talk about why this is kind of concentrated in the top right corner, because you would expect to see something more hyperbolic like that, and that's also a possibility. But this sounds really neat so far, but it could be possible that this is just the product of a few kind of simulations. So how rare are these outcomes? So we found that if the secondary parasite was introduced before the deterministic threshold, which is the 10th or 12th day of infection, depending on the thermal performance curve you're using, up to 71% of simulations resulted in longer periods of co-occurrence relative to that baseline. But even if that secondary parasite was introduced after that deterministic threshold, so deterministically, first parasite would already be out of the system, or the immune system would be so large that it wouldn't be able to invade in the first place. But once we consider stochasticity, we find that up to 46% of simulations still show some period of co-occurrence with the initial infection. And that can also be expressed by thinking about how much environmental variability extends that invasion threshold. So before, we're seeing 10th to 12th day of infection. Once we consider environmental stochasticity, we see that we can extend that invasion threshold by quite a bit. Okay, so this is all thinking at a within host level, but we're often interested in the among host level for a very good reason. And normally we'd expect single infections to be much more transmissible than multiple infections, given that single parasites don't have to compete with anybody else. But we found that among limited cases, environmental variability can increase the transmissibility of the secondary parasite relative to a single parasite suggesting that there's some sort of sweet spot in which environmental variability can both maintain co-occurrence and increase transmissibility. I think by decreasing parasite population abundances to levels that elicit a smaller immune response, allowing both of those parasites to kind of exist within their host for some longer period of time. Okay, so the main takeaway here is that a variable environment has the potential to increase the ability of a secondary parasite to invade a system in which a single parasite already exists, promoting longer periods of co-occurrence between parasites and potentially, in some cases, also increasing the transmissibility of co-occurring parasites. And I think there are some important real-life implications here, despite the fact that I have not talked about a real system yet. So, for example, amphibians are experiencing disease-mediated declines, and it's been posited that climate change can exacerbate the effects of disease on the system by impeding immune functioning, leaving them more susceptible to disease and less able to fight disease. But considered alongside our findings, we suggest that climate-mediated immune suppression may, yes, prevent those amphibians from mounting a sufficient immune response, but that lack of sufficient immune response might also increase the viability of multi-parasite infections, enabling longer periods of co-occurrence among these parasites and maybe affecting disease outcomes. It's additionally been posited that internal temperature variation may aid in bats' immune responses. I think it would be interesting to actually take a closer look at, again, how those internal temperature variations affect the parasites that they host, in addition to their immune responses. And if you want to read the paper, you can scan this QR code, which I forgot to mention at the beginning, so now there's not that much time. Okay, and I'm on to project 3. Okay, so as I've been harping on about for way too long at this point, host parasite dynamics are often mediated by temperature, and we can think about how they do that in a number of different ways. For example, by affecting vital rates, and we can also think about at these higher levels of organization affecting where hosts even are in a system. And bless you. So climate change is important because it may impact transmission rates and affect if and when epidemics can emerge, which brings up this really important question. If we expect more epidemics to emerge as a consequence of climate change, is there a way for us to predict the emergence of those pandemics? And I'm not the first person to ask this question, and it kind of comes back to bifurcation theory. So a lot of people think about ecological systems as being characterized by critical thresholds. If we think about these systems as dynamical systems, they should have critical thresholds. So when parameters change, the system may be forced through a local bifurcation and experience what we call a critical transition, which is important because it changes the stability properties of the system. And as the system approaches that local bifurcation, as the parameter changes, the rate at which the system recovers from small perturbations to the system, so environmental change, for example, slows down. And this kind of slowing down effect leaves behind statistical signatures termed early warning signals, and I'll talk about some of those in a sec. But importantly, disease systems can also experience critical transitions when basic reproductive number, R naught, increases from less than 1 to greater than 1. And just as a reminder, R naught is the number of secondary infections arising from a single infection in a completely susceptible population. And we would call this a transcritical bifurcation, which a lot of people think is uninteresting, but transcritical bifurcations can be interesting if you're very interested in disease, like I guess I am. So while studies of different systems have yielded kind of wildly varying results, the absence of control populations within which subepidemic spread fails to transition into epidemic spread really limits our ability to interpret our results. And I think that concern is really well summarized in this paper that Alan's on in the middle here, Early Warning Signals and the Prosecutor's Fallacy, if you're interested. Okay, so when I was at U of T, I very briefly overlapped with another PhD student, Devin Kirk, who's great. And he wanted to figure out if he could develop a mechanistic model to predict the temperature at which an epidemic would emerge in a Daphnia system. And he did a pretty good job because he got Employee of the Week every week for like 4 years, I think. So it's probably still in the lab today. So Daphnia are freshwater planktonic crustaceans, and in nature, they host a wide array of parasites, including mycosporidium parasites. And Daphnia become infected when they passively ingest the parasite while grazing. That parasite infects gut epithelial cells and replicates into clusters of spores, which after some period of time eventually burst, releasing new spores into the environment and also into the Daphnia to infect new cells. When they're released into the water column, they can infect new hosts by ingestion. Yeah. Okay, so here's Devon's model. It contains 6 temperature-dependent parameters, which I've highlighted in blue. And it tracks the number of susceptible hosts, which grows via immigration and recruitment and declines via infection, death, and harvesting for the experiment. The number of infected hosts is also tracked, and this number grows by immigration and infection. So we're putting in some infected hosts as well, and declines via death and harvesting. We also track dead hosts because they can transmit as well. And we are also tracking the number of environmental spores in the water here, which are shed by both dead and infected hosts. So he used this model to derive R naught and then used this equation for R naught to predict the temperature at which an epidemic could emerge in the system. And all the parameters here were empirically estimated with an experiment that took place before this one. But he ended up estimating that temperatures of 12 degrees Celsius and above would enable epidemic emergence in this system. So to test this prediction, we embarked on a 120-day experiment involving 8 Daphnia populations and their mycosporidium parasite. So populations 1 through 4 in blue here are held at some constant subepidemic temperature of 10 degrees, while populations 5 through 8 are subjected to a warming treatment where the temperature of the system is increased by ½ degree between 10 degrees Celsius and 13.5 degrees Celsius over that 120-day period. And the simulation results seem to line up pretty well with the experimental results. If the temperature of the system exceeds 12 degrees Celsius, we should see epidemic emergence, which is really, really cool. But building these trait-based models to predict the conditions under which an epidemic may emerge is really, really data intensive and requires a lot of work that we don't always have the time and resources to do, especially when we're concerned about epidemics. So we wanted to know if we could use early warning signals to predict this transition from a subepidemic to an epidemic state. So makes it look like it only required 2 steps. When I did it, it felt like more than 2. But at first, we used the simulated and experimental data to calculate 10 statistical metrics within sliding windows that we expected to see some difference in if we were approaching a critical transition. And then we measured trends in each of those statistics over time using Kendall's tau, which I'll just refer to as the trend coefficient because it's easier to say. So to explain Kendall's tau a bit, let's say that we have prevalence data over some period of time, so the prevalence of disease population. So first, we calculate our metric of interest within some sliding window, let's say 5 days, so days 1 through 5. And then we move on and calculate that same metric of interest over the next sliding window, so days 2 through 6, and 3 through 7, and so on. And that gives us some relationship between time and here variance. And what we're interested in is the ordinal association between that metric and time. And that's what we use Kendall's tau to calculate. So as a metric, we can consider Kendall's tau to be some value between -1 and 1. And if there's perfect correlation between the value of a metric of interest and time, we should see a Kendall's tau of either -1 or 1, depending on the direction of that relationship. So for example, if we do all this work and the resulting trend coefficient is positive, we might cautiously conclude that an increase in a metric over time is predictive of some critical transition that's impending. And if we have X number of time series, we can calculate X number of trend coefficients and plot them as distributions to compare control and test populations, which is what we want to do here. And we can even take it a step further and summarize the overlap between these distributions and interpret that overlap as the probability that a randomly chosen trend coefficient is larger, or sorry, that a trend coefficient is larger than some randomly chosen value. So these are the metrics that we took a look at. This slide is a lot to take in, but I wanted to include it in case people had questions. I've highlighted in red the kind of common metrics that people are really interested in. Specifically, people are really interested in change in variance as you approach a critical transition, again, because the system becomes less readily able to move back to its equilibrium after being disturbed. So we would expect to see an increase in variance, for example. So after analyzing the simulated time series, we summarized our findings using these density plots. So again, we have these kind of 10 metrics of interest corresponding to 10 density plots here. And each density plot is showing you 1,000 control trend coefficients and 1,000 warming coefficients in blue and pink, respectively. And I've highlighted mean prevalence here to show you that among control populations, the average trend coefficient sits around 0. But if we look at warming trend coefficients, the average coefficient sits at about 0.4, which is higher than 0. So we might conclude that an increase in mean infection prevalence would predict an oncoming epidemic emergence. But we don't really see what we expected to see anywhere else. So let's take a look at the experimental data. So here, each star is an experimental population. So we have 8 stars per metric. And the metrics are shown on the horizontal axis here. And we have value of the trend coefficient on the vertical axis. And that dotted line in the middle denotes the trend coefficient of 0, where we see absolutely no correlation between a metric of interest in time. So here we see a similar difference in mean prevalence between control and warming populations. But we also see differences in variance in the index of dispersion in control and warming populations, which I've highlighted in green here. But if we take a closer look inside these purple bubbles, you'll see that in a couple control populations, we're getting these really, really high trend coefficients that are kind of rivaling the values of the warming or test populations, which is unexpected and weird. So to quantify our confidence in the experimental findings, we used 4 different methods to generate null distributions of the trend coefficient, and then used those null distributions to calculate the probability of observing the true trend coefficient by chance. And here's what we found. So these are density plots again, with the trend coefficient on the horizontal axis and density on the vertical axis. And we're looking at trends in mean prevalence, variance, and the index of dispersion across all 8 experimental populations. So the top 4 are the test or warming populations, and the bottom 4 are control populations. And here we're just comparing the true trend coefficient, which I've shown as a blue or red dot, to the 95th quantile of each of these 4 null distributions. And those quantiles are shown as little black lines, which are likely a bit too hard to see. And the null distributions are largely overlapping, but they're denoted by some different colors here. And we can talk a little bit about why the pink one looks so different if you want to later. But if we focus in on an example here, we can see that in population 7, the true trend coefficient for mean prevalence in red is more extreme than 95% of values included in each of the 4 null distributions. So we might conclude that this is a reliable detection. And here I've starred every case in which the true trend coefficient is more extreme than the 95th quantile of each of the 4 null distributions. And importantly, you'll notice that we're seemingly just as likely to detect these signals in control populations as we are to detect them in warming populations, which is a big problem. So that's a problem because it looks like false positives might be confounding our interpretation of early warning signals in pre-epidemic systems, which calls into question their reliability as indicators of at least climate-mediated epidemic emergence. And even more notably, the simulation results suggest that more robust simulation results can be as robust as you want. You can have a million time series. And the fact that we don't see what we expect to see here suggests that more robust sampling regimes might not solve this problem. But differences between the simulation and empirical results also do suggest that there is still value in robust and long-term disease surveillance programs, especially if we want to avoid these kind of false positive results. So here we, I think, identified some pitfalls in the detection of early warning signals in disease systems, especially characterized by environmental transmission. So most cases in which I've seen people looking for early warning signals in disease systems, those systems have been characterized by either direct or vector-borne transmission. And I'm not quite sure why it would be different in environmental systems. But resolving this difficulty seems important, especially in the face of increasingly frequent extreme climate events, which reduce access to clean water and increase the transmission of diseases like cholera. We've seen a lot more cholera deaths in the last number of years. So that's project 3. And I just very, very quickly in the last couple of minutes wanted to briefly talk to you about what I'm doing now. So like Marissa said, I arrived in September and I'm starting work on a couple different projects that I'm really excited about. I'm moving from the kind of early warning signal region. I'm staying within regime shifts a bit. I think they're quite cool. So I'm interested in if we can predict and differentiate between different types of regime shifts. So bifurcation-induced tipping points versus rate-induced tipping points versus things like transient states. And I'm especially interested in when the differences in our ability to predict these regime shifts, when the differences between them are actually relevant to ecologists and conservation biologists. So we're often interested in predicting these shifts in imperiled systems, but it's unclear what that information would allow us to do, I think, and how we think about these things is also really convoluted. But additionally, and sort of related to some of the disease work I've talked about here, I've also been really interested in the impacts of conservation management on disease spread. So I'm thinking a lot about how management interventions might affect disease risk in conservation relevant systems. So if you have thoughts on any or all of these things, or the other stuff that I talked about, I'd be very happy to chat. But yeah, these are people that I have to thank and funding sources. So thank you for listening.
I have kind of a naive question as somebody who's never really fitting models to data. In the first part, where you talked about the issues related to the fact that you could essentially correctly recreate the dynamics with the wrong underlying model.
Yeah.
And I guess I would be interested in hearing a bit about why that's a problem or for what goals that would be a problem, because, you know, like, again, very naive, but
No,
it's like, well, if it's recreating the dynamics, like, do we really care? Or when would we care? For what purposes that the underlying?
Yeah. So no, I don't think that's a naive question. I think right dynamics, wrong underlying model is important because if you have the wrong underlying model, this is also assuming that you can identify the like true mechanistic model, which is like maybe more of a philosophical question. But like if you have the wrong model, then there's some set of dynamics that some model can produce, and it's different than some set of dynamics that a different model can produce. So even if there's some overlap, like say 2 different models can both give you a stable limit cycle, but the other things they can give you are different. So if you have the wrong underlying model, you might expect, well, this system is currently doing this, but it could do any of these things in the future if parameters X, Y, or Z change. And that kind of conclusion of what is possible would be different depending on the underlying model that you're thinking about. Does that answer?
Yeah.
Yeah.
I got 2 questions on part 2. You were very careful about saying co-occurrence rather than coexistence because you're looking for finite time scales, and like the work of Peter Chesson and others was thinking about infinite time scales, right? So, but nonetheless, like that sort of modern coexistence theory approach, one of the things they try to do is say, what are the underlying ecological factors that are determining whether things coexist or not? And so I was wondering, did you look into like, what are the maybe underlying mechanisms or factors that might be leading to longer versus shorter co-occurrence? Like it seems like you have a relative nonlinearity occurring in the model because the immune system has that sort of type 2 response.
Yeah.
So variability in the like parasite numbers is going to lead to a diminishing effect of the immune growth. So I don't know, did you look like into like which of those aspects of that system are most important? Is it that sort of Jensen's inequality, or is it more just the variability leads to a longer time sometimes before the parasite that's initially introduced actually starts getting to large enough numbers to sort of kickstart the immune system?
Yeah. Yeah. So you're right that we focused on relative nonlinearity. It seemed maybe the most obvious choice thinking about environmental variability. So that's, I think, why we went there initially. But I like this paper, but I also hate it because, I mean, like any paper, this just, we took a model and did a bunch of stuff with it. You could do a million more things. And those million more things are obviously haunting. Like something that I think would be really interesting to think about is like how like spatial separation regarding like where infections happen in a host could affect these kinds of outcomes. Because you could imagine like different types of parasites prefer to stay in different types of tissues. And if you have that kind of like spatial separation, you might get something totally different where they're able to coexist for way longer, or if two different parasites exploit different parts of the immune system. So like here we kind of assume because there's a learning time that we're thinking about an adaptive response, but there's also an innate response, and which parasites trigger adaptive because you've experienced the infection before versus innate because it's novel could also create a kind of difference in time scale for how that reaction of the immune system happens. So these are things that I would love to think about, but would have made the paper much longer, I think.
Sure.
But yeah, those are kind of my initial thoughts there.
My second, if I can ask a second question, for the third part, we're looking for early warning signals for a transcritical bifurcation,
Yes.
which is more of a localized bifurcation. While a lot of the like initial work on sort of early warning signals were for like saddle node bifurcations, which are more of a global bifurcation that have big effects. So I'm wondering how much of the difficulty of finding a good early warning signal is because you're looking at transcritical bifurcation rather than a saddle node. Like, is that the real underlying cause of making it harder to see something's going to change?
Yeah, that's a good question. I feel like it's hard because, like I mentioned, all of the kind of previous work looking for early warning signals of epidemic emergence, and of which there's a lot. I think almost all of that work says they do find early warning signals, but they're also looking at observed data, usually over very long time periods, or in systems that we have like really good understanding of. Like I'm thinking of there's a paper looking for early warning signs of malaria outbreaks, and those are kind of like recurrent outbreaks, and they have data going back to the 80s. And those outbreaks are maybe easier to track because people are coming into a hospital. It's like very identifiable symptoms. So you're getting a lot of certainty in the case counts. Well, as much certainty as you can have in case counts, maybe. So we do see like in cases like that, they're allegedly easy to find. So I'm thinking that it might not be the transcritical, like it might be harder relative to something like a Hoff bifurcation, but I don't know if it's like prohibitive. So I was thinking here, like especially since we didn't see them in the simulated populations, like not seeing them in 8 experimental populations is kind of whatever, but not seeing them in the simulated populations, I think was eyebrow raising to me. And yeah, like you could think about, like I think during my defense questions came up about like the size of the population just might not be big enough, like especially in the experimental populations. Infection prevalences were taken from like subsamples of the population. So you're seeing prevalence counts kind of oscillate between like 0, 1, and 2, 0, 1, and 2. And like, if you're only sampling for, I mean, 120 days is a long time to run an experiment, but it's not actually a very long time series. So those kinds of limitations could have been really impactful, I think, on the detection. But I am also interested in kind of the mode of transmission as well, especially if you're looking at like real empirical data from a real system, not in a lab. So where like source differs and detection differs and things like that, then the data could get really messy in a weird way. But yeah, does that answer your question?
Sure.
Okay.
Thank you.
Let's thank our speaker.